Hello, my name is Joshua Gabi. I welcome you to Cell Nucleus. What are cells with a delimited nucleus called? What are the main elements of the nucleus? Cells with limited nucleus are called eukaryotic cells. Organisms composed of one or more eukaryotic cells are called eukaryotes. The main elements of the nucleus are the chromatin, made of DNA molecules, the nucleolus, and the karyolymph or nucleoplasm, and the nuclear membrane, or what we call the karyotheca. Do all eukaryotic cells have nucleus and only one nucleus? There are eukaryotic cells without a nucleus and others with more than one nucleus. Osteoclasts. These are cells responsible for resorption of the osseous matrix. For example, are multinucleate cells. Striated muscle fibers are multinucleated cells too. Red blood cells are examples of enucleated specialized cells. Of which substance is chromatin made? Chromatin is made of DNA molecule associated with proteins called histones. What are heterochromatin and euchromatin? Chromatin is uncondensed nuclear DNA. The type of DNA morphology in interphase. The phase of the cell cycle in which the cell is not dividing itself. In this phase, the cell cycle chromatin can be found as heterochromatin, more condensed and dark in electron microscopy. Portion of DNA molecules and has euchromatin, less condensed, lighter portions of DNA molecules. Since it is uncondensed, eurochromatin is the biologically active portion of the DNA. That is the region that has active genes to be transcripted into RNA. The heterochromatin represents the inactive portion of the DNA molecule. What is the relationship between the concept of chromatin and chromosome. Are euchromatin and heterochromatin part of chromosomes? Every flat filament of chromatin is a complete DNA molecule, a complete double helix, that is, complete chromosome. A DNA molecule may form euchromatin or heterochromatin portions. Thus, both are part of chromosomes. In the phase when the cell is not dividing, interphase, is there activity within the cell nucleus? In the interphase, there is intense metabolic activity in the cell nucleus. DNA is duplicating, euchromatin is being transcripted, and RNA is produced. How are the concept of chromosome, chromatin, and chromatids related? In which phase of the cell cycle does DNA duplicate? Chromatin is a set of filamentous DNA molecules dispersed in the karyoplasm, forming euchromatin and heterochromatin portions. Each chromatin filament is a complete chromosome, that is, DNA molecule, a double helix. The chromatin of the human somatic cell is formed by 46 DNA molecules. Those are 22 homologous chromosomes and one pair of sex chromosomes. In interphase, the cell prepares itself for division and duplication of DNA molecule occurs. The duplication of every DNA molecule forms two identical DNA double helix bound by a structure called centromere. In this phase, each identical chromosome of these pairs is called chromatid. It is also during interphase that the chromatids begin to condense, assuming thicker and shorter shape, typical to chromosomes illustrations. So the phase of cell cycle in which DNA duplicates is interphase. Some biology textbooks call chromosomes a unique filament of chromatin, as well as the condensed structure made of two identical chromatids after the DNA duplication. Rigorously, the pair of identical chromatids bound in the centromere or are two copies of the same chromosomes, and therefore there are two identical chromosomes, not only one. What is the structure that maintains identical chromatids bound? The structure that maintains identical chromatid bound is the centromere. How is the chromosome region where the centromere is located called? How are chromosomes classified in relation to the position of their centromere? The chromosome region where the centromere is located is called primary constriction. In microscopic view, the region is narrower or stricter than most of the part of the chromosome. According to the position of the primary constriction, the chromosomes are classified as telocentric, acrocentric, submetacentric, or metacentric. What are the primary and secondary constriction of a chromosome? What is the other name given to the secondary constriction? Primary constriction is the narrower region of the condensed chromosome, where the centromere, the structure that unites identical chromatids, is located. Secondary constriction is a region similar to the primary constriction, narrower than the normal thickness of the chromosome 2. In general, it is related to genes that coordinate the formation of the nucleolus and control the ribosome RNA synthesis. For this reason, the secondary constriction that can be one or more in chromosome is called nucleolus organizer region, NOR. What is homologous chromosomes? Which are the human cells that do not have homologous chromosomes? Chromosomes contain genes, genetic information in form of nucleotide sequences that command the protein synthesis, thus regulating and controlling the activities of the cell. In the nucleus of somatic cells of diploid being, every chromosome has its corresponding homologous chromosome, both containing alleles of the same gene related to same functions. This occurs because one chromosome of one pair comes from the father and another one comes from the mother of the individual. The chromosomes that form a pair with alleles of the same gene are called homologous chromosomes. In humans, there are 22 pairs of homologous chromosomes, plus one pair of sex chromosomes. The sex chromosomes are partially homologous. The only human sex that do not have homologous chromosomes are the gametes, since during meiosis, the homologous chromosomes are separated. What is the difference between the concept of karyotype and the genome? Genome is the set of DNA molecules that characterize each living being and each species. The concept then includes specific nucleotide sequence of the DNA molecule of each individual or species. Karyotype is a set of chromosomes of the individuals of given individual or species concerning morphology and the number of each chromosome or pair of homologous. Can two normal individuals of the same species with the sexual reproduction have identical genome and identical karyotypes? How is human karyotypes usually represented? Except for clones, individual created from nucleus transplantation, like the doll sheep, and homozygotic twins, it is very improbable the genome of two individuals of the same species and generated by sexual reproduction to be identical. Nevertheless, the karyotypes of two normal individuals of the same species and of the same sex are always identical. The human normal karyotype is represented by the formula 44 plus XX, that is for women, and 44 plus XY for men. What is the other name given to the sex chromosomes? What is the function of the sex chromosomes? Sex chromosomes are also called allosomes. The other chromosomes that are not sex chromosomes are called autosomes. Sex chromosomes get such a name because they have genes that determine the sex, male or female, of an individual. Sex chromosomes also have genes related to other biological functions. How many chromosomes does a human normal haploid cell have? How many chromosomes does a human normal diploid cell have? How many are sex chromosomes within each of them? The human haploid cell is the gamete, that is the egg cell and the sperm cell. The human gamete has 22 autosomes and 1 allosome, i.e. 23 chromosomes. The diploid cell is the somatic cell, and it has 44 autosomes and 2 allosomes, that is 46 chromosomes. Gametes have 1 sex chromosome and somatic cells have two sex chromosomes. Do phylogenetically proximal species have cells with proximal chromosome counts? The number of chromosomes typical to each species is proximal for phylogenetically proximal species. For example, orangutan, gorilla, chimpanzee, and human. But it is not impossible that evolutionarily distant species like a rat and oat bear similar karyotypes and the same total number of chromosomes. Even presenting equal number of chromosomes evolutionally, distant species have radically different characteristics, since the quantity and the sequence of nucleotides that compose their respective DNA molecules are quite different. What is the nucleolus? The nucleolus is a small, optically dense region in the interior of the cell nucleus. It is made up of ribosomic RNA and proteins. One nucleus can have one or more nucleolus. Of which structure is the nuclear membrane composed? Eukaryotic cells have nucleus delimited by two juxtaposed membranes that continue with the membrane of the endoplasmic reticulum. The nuclear membrane, or karyotheca, presents pores through which substances pass. There are also ribosomes adhered to its external surface. Thank you very much for participating in Cell Nucleus. Please do like, share, and subscribe.